Hello, I'm Anas Al Ubaidi, and in this video, we will be covering the first session or first topic or first video in chapter 9, as we will be starting with chapter 9 as part of the whole syllabus or curriculum of the uh, from the Advanced Medical Science Liaison or MSL and Medical Monitor MM certification course. For short, it's AMSLC by the CCRPS. And chapter 9 that we will be starting with through this video is about clinical trial phases and procedures. And the topic for this video in this video is about the clinical trial monitoring procedures phases 0 to 4. Introduction. Clinical trial monitoring serves as the backbone of clinical research, ensuring that ongoing trials across all phases 0 to 4 maintain the highest standards of integrity, safety, and validity. So, one of the most important applications in order to maintain the highest standards of integrity, safety, and validity in clinical research is done by clinical trial monitoring, by the monitoring. And by doing so, the highest standards will be maintained for each integrity, safety, and validity in clinical research. This complex process is essential for safeguarding participants while producing reliable data to support regulatory approvals and future medical breakthroughs. Meaning, this complex process, it's the monitoring, the clinical trial monitoring, the monitoring overall that is done by medical monitor or any other monitor is important, is essential, is crucial in safeguarding participants or the patients who are participating or the subjects while producing reliable data, proper, accurate data to support regulatory approvals and future medical breakthroughs. Now, monitoring tracks everything from protocol compliance to quality data generation, ensuring all activities adhere to good clinical practice, the GCP, and ethical guidelines. So the monitoring tracks all the aspects in clinical research, including protocol compliance to generation of the quality data, ensuring all activities adhere to good clinical practice, GCP, meaning making sure tracking that the GCP is being applied and the ethical guidelines as well. The structured progression of clinical trials through phases 0 to 4 plays a vital role in the evaluation of the new drugs, treatments, or devices. Each phase builds upon its predecessor, focusing on specific objectives. So each phase has its own objective, from initial drug behavior assessments in phase 0 to large scale efficacy analysis in phase 3, and real world safety evaluations in phase 4, as it is called the post marketing phase, phase 4. Advanced monitoring procedures tailored to each stage provide oversight tailored to the evolving complexity of trials. For instance, phase 4 involves microdose tracking, while phase 3 monitoring emphasizes multi center consistency across thousands of participants. And this is as an example, the difference, for instance, between phase 4 and phase 3. Ensuring patient safety and high quality data requires constant vigilance in the face of challenges such as managing vast datasets, handling varying global regulations, and ensuring uniformity across sites. So, as any application for any aspect in clinical research or any uh, um, advancement or any um, procedure in clinical research, we will be facing challenges. While obstacles remain, recent technological innovations like artificial intelligence, AI, electronic data capture, EDC systems, or electronic case report forms, ECRF, and centralized monitoring platforms have transformed how clinical trials are monitored, making oversight more precise and efficient. So here applications of technology, as for examples AI or EDC and the centralized monitoring platforms, have transformed how clinical trials are monitored, meaning um, upgraded the monitoring procedures, the monitoring application and aspect into more advanced levels, and by doing so, in order to make oversight being more precise and efficient, which are the outcomes for monitoring that we are looking for. Key points for the topic for the day. We'll start with number one, overview of clinical trial phases. Phase zero, which is called exploratory studies. Now, purpose of phase zero, establish whether a drug interacts with the human body as predicted by preclinical studies. So phase zero here, exploratory studies, is more like preclinical studies. Participant sampling here will be fewer than 15 participants who receive microdoses that are pharmacologically insignificant but sufficient to study the initial behavior. So as we see here, the sampling, the number of the participants, the amount of the sample is very small as it is for preclinical studies purposes, exploratory studies as it's called, phase zero. And here it is done by microdosing. However, microdoses that are pharmacologically insignificant, then why? The reason why be- it is insignificant, however, but sufficient to study the initial behavior. So the purpose here is to study the initial behavior uh, through insignificant microdoses for small amount of sampling, small amount of participants or subjects. And here it is called phase zero exploratory studies as preclinical studies. Now the focus here, understanding pharmacokinetics, for example, how the drug is absorbed and distributed. So the pharmacokinetics of the drug, of the new drug, or study medication or SM or investigational product IP, as to study the absorption, the distribution, the metabolism, and the excretion of it. So here, the pharmacokinetics. Evaluating pharmacodynamics as well, and the pharmacodynamics meaning the effects on the body, how the body will react, what are the signs and symptoms of what that will be shown by the body of the participants or through the body of the participants. So evaluating pharmacodynamics, for example, the drug's effects on targeted cells or tissues. So the changes that will be shown in the body, in the tissue cells of the body of the participants, pharmacodynamics. Example, testing an oncology drug to determine whether it penetrates tumor cells effectively prior to, before progressing to phase one. Phase one, safety and dose escalation. So it's about safety. Purpose, identify the maximum tolerated dose. So phase zero, the exploratory studies was microdoses. It was microdose. Now the maximum tolerable dose, MTD, and assess short-term adverse effects. So safety, the safety of the medication through the using maximum tolerable dose, not microdose like phase zero. Now participant sampling also small, but little bit bigger than phase zero, but still small, 20 to 80 healthy volunteers or patients, depending on the drug safety profile and purpose. So 20 to 80, minimum 20 to 80 patients or participants or volunteers or subjects. Focus testing dosages, routes of administration, for instance, oral versus intravenous, IV, and ensuring basic safety. So the safety, the route of the administration, whether oral or intravenous or even intramuscular, so safety, the maximum tolerable dose, and the amount will be small but bigger than phase zero. Example, exploratory trials adjust doses of an anti-inflammatory drug to monitor liver enzyme levels and pinpoint safe ranges. And this is an example for phase one. Phase two, efficacy assessment. Now, preclinical studies for phase zero. Phase one was the safety. And phase two, here the efficacy. Assessing the efficacy. The purpose determine efficacy in treating the target condition while closely monitoring side effects. So side effects also, safety and side effects started from phase one, and here efficacy and side effects monitoring as well in phase two. Participant sampling several hundred, so bigger. Now the more advanced phase we go to, more advanced phase we apply, the bigger sample will be in compared to the prior phase. So several hundred patients with the disease or condition being studied. So diagnosed with the condition or diagnosed with a disease that is targeted through the study medication or the investigational product. Focus evaluating treatment outcomes such as symptom reduction or disease remission. Example, measuring lung function improvement in asthma patients receiving a new respiratory therapy. Phase 3, large scale efficacy and safety. So, safety and efficacy like phase 1 and phase 2 and phase 3, but larger scale in compare to phase 1 and 2. And here, both in phase 3, adding the purpose or the goal or the determination of both phase one and two, safety from phase one and the efficacy from phase two and phase three, as we have larger scale efficacy and safety, but we'll always having starting from phase one, we'll always having the monitoring of the side effects as well or adverse events. Now, the purpose for phase three, compare the new intervention against the standard of care or placebo while evaluating long-term safety. So here we'll be using placebo or the standard of care. Standard of care meaning the targeted disease or the condition. We have actual medication for it, choice medication of choice or treatment of choice that is already in the market, already being used, already being prescribed. So we use it against, or we use the study medication or the investigational product or new medication against that already prescribing medication, or using the study medication or the investigational product against a placebo. And the evaluation will be long-term evaluation. Participant sampling, as we see here, bigger in compared to the previous phases, as the participants or the subjects or the patients will be 300 to 3,000 or even more. Randomized into various groups. And here we'll use randomization, whether single blinded or single masked randomization or double masked or double blinded randomization. As we'll be using again the investigational product or the study medication against the placebo or against, if not the placebo, against already standard of care medication or the medication of choice that's already being prescribed and used for the targeted medicine disease or condition. The focus, larger scale studies to assess therapeutic superiority, equivalence, and side effects profiles. Example, a multinational oncology trial compares survival rates between an experimental immunotherapy and an existing chemotherapy protocol. So here, see the example. Experimental immunotherapy, which is the investigational product, let's call it, or the study against existing chemotherapy protocol. Phase 4, post-marketing surveillance. So the purpose, monitor long-term safety also and the effects. So also we'll go back in phase 4 into monitoring, so doing a surveillance on the safety and the adverse events or the side effects of the medication that is approved to go marketing, and now we'll start phase 4 as post-marketing surveillance. And by doing so, monitoring again the long-term safety and the effects of the intervention after it has received the regulatory approval. Participant sampling thousands because it's in the market, it's being used to the patients, for the targeted disease or the condition. So thousands, even bigger, more bigger sampling than the previous, in compared to the previous phases. So thousands of participants monitored over years, long term, to track real world side effects or emerging benefits. Focus observing rare adverse events and new indications or long-term risks and cost analysis. Example, identifying cardiovascular risks of a widely prescribed diabetes medication 5 years after its approval, as is post-marketing surveillance, which is phase 4. Here, a nice diagram of drug development phases. We'll have exploratory studies, initial participant testing as a phase 0, 15 patients, and then into 80 for phase 1 trials, looking for the safety and dose escalation testing. Number 2, advanced monitoring strategies. So general objectives for monitoring: ensure strict adherence to protocols and GCP, good clinical practice, protect participants, patients by monitoring for adverse events in real time, maintain data quality by identifying and addressing inconsistencies or missing records. Phase-specific monitoring strategies. We have phase 0 will be centralized small-scale oversight supports NICM pharmacodynamic evaluations. So pharmacodynamics. Example, tracing microdose effects, because we said in phase 0 we have microdose that is pharmacologically insignificant. So tracing microdose effects using centralized pharmacology databases. Phase 1, daily participant review ensures immediate detection of dose toxicity, as for an example, monitoring liver enzyme shifts in healthy volunteers receiving escalating drug doses. Phase 2, frequent site visits and subject reviews bridge efficacy, analysis, and safety compliance. So frequent on-site monitoring visits and subject reviewing a bridge efficacy analysis and safety compliance. As for example, leveraging patient-reported outcomes to track remission rates over treatment cycles. Phase 3, multi-center monitoring, focuses on global consistency, randomization, integrity, and blinding. Example, deploying EDC, electronic data capture systems, to compare data from different countries in a cardiovascular trial. Phase 4, passive yet structured observation through post-market data analysis, healthcare provider feedback. So post-marketing surveillance, post-marketing data analysis, and it will be for long term. So passive yet structured observations from post-market data analysis, healthcare provider feedback, and patient registries as well. Example, centralized reviews of post-marketing adverse events flagged by pharmacovigilance systems or PV systems. Monitoring tools and techniques. We'll have here electronic data capture systems or electronic case report forms, ECRF, streamlines data accrual by integrating laboratory, site, and reporting results. Example, rapid flagging of outliers in multi-center trials using real-time alerts. Interactive response technology, IRT. IRT, as it's short for interactive response technology. Interactive response technology. One of the tools or techniques that the monitors are using as IRT or interactive response technology automates drug inventory tracking, patient randomization, and recruitment updates. Example, ensuring drug shortages at specific sites are addressed promptly, instantly. Remote monitoring, as how there are, we have on-site, in-person, physical remote monitoring. We have the remote monitoring as well, distance, like through Zoom meeting and such, enhances oversight via real-time access to patient records, reducing reliance on site visits. Example, virtual meeting of decentralized trials targeting remote populations. Here, a nice diagram about clinical trial monitoring process as we have the starting from the comprehensive trial oversight or starting from protocol adherence, ending with comprehensive trial oversight. And we have here starting with phase 0 monitoring, phase 1 monitoring, phase 2 monitoring, phase 3 monitoring. And of course, then we have phase 4 monitoring, which is post-marketing surveillance. Number 3, we have the challenges. Now, the challenges we might face in the clinical trial monitoring, data volume management. The data volume as one of the challenges. Issue here, increased trial complexity leads to overwhelming data sets. Solution, scalable digital platforms prioritize abnormal or high-risk signals for initial review. We have B, regulatory variation. Issue, diverging regulatory guidelines across countries create delays. And if we go back here to the diverging regulatory guidelines across countries, here we go back to the idea we have previously and through our previous sessions explained about some of the differences in the regulatory sections or in the protocol, some minor differences or some small differences in between the sites who are conducting the same exact trial or study due to the local regional regulations and laws. So same idea here. Solution, engage local experts early to establish regional compliance frameworks. And always the solution will be the same, is to engage with, involve the local experts. See, limited resources issue, budget constraints hinder robust on-site site assessment. Solution for it will be hybrid monitoring. The distance for and physical, in person. Hybrid monitoring models balance cost and efficiency by maximizing digital resources. And by maximizing digital resources. So the hybrid alongside applying the digital resources like EDC, IRT, and such. Risk-based monitoring RBM complexity issue, misidentifying or overweighing some risk factors lowers RBM precision. The risk-based monitoring precision. Solution for that, use predictive AI, artificial intelligence models to refine risk driver detection. Here, a nice diagram about the challenges, about the clinical trial monitoring challenges, as we have the data volume management, limited resources, regulatory variation, at RBM, risk-based monitoring complexity. Now, we have recent advancements in clinical trial monitoring. So, despite of the order, we'll start with number 1, centralized monitoring consolidates site-specific and patient data for quick real-time review. Example, flagging delays in pharmacovigilance reports, PV reports across Eastern Europe. Machine learning algorithms, and here detect unexpected patterns or anomalies across large datasets. Example, highlighting unusual cluster trends and adverse reports for accelerated investigation. Predictive risk analytics anticipates potential recruitment issues or dropout risks early on. Example, adapting resources for underperforming recruitment centers during a phase 2 immunology trial. Blockchain technology, the audit trail system, ensures unmodifiable records and enforces accountability. Example, tracking consent forms in decentralized trials spanning diverse regulatory environments. So tracking of data. Here, a nice diagram summarizes the recent advancements in monitoring, in clinical trial monitoring technologies. We have the centralized monitoring, site-specific data, patient data, um, blockchain technology, the audit trail, unmodifiable records, accountability enforcements. We have the machine learning algorithms, but pattern detection, anomaly highlighting, predictive risk analytics, recruitment issues, and dropout risks, and all within recent advancements in clinical trial monitoring. Applications in practice as starting from phase 0 to 4. So phase 0 application example, we have microdose tracking for pharmacokinetic analysis, and pharmacokinetic here meaning the absorption, the distribution, the metabolism and the excretion for the medications. And here, for short, ADME or ADME for abbreviations, A as for in order, in the same order, A for absorption, D for distribution, M for metabolism, and E for excretion. So the pharmacokinetic analysis through microdose that pharmacologically insignificant for phase 0. Phase 1, monitoring incrementally rising toxicities. Safety, right? Phase 2, analyzing remission consistency across sites, the efficacy. And phase 3, both safety and efficacy, but on bigger sampling and amount and compare, evaluating datasets across multi-center trials. And phase 4, pharmacovigilance, PV for rare adverse events, as it is poor marketing, post as it is post-marketing surveillance. Through the detailed application of these principles and tools, clinical trial monitoring remains a critical safeguard for achieving ethical, scientifically valid, and impactful research outcomes globally. We have summary tables to summarize what we have explained in this video as in clinical trial phase overview. We have the phase, the purpose, participant sampling, unique focus, and example. Phase 0, uh, purpose is test drug behavior, initial behavior at microdose levels that are uh, pharmacologically insignificant. Sampling about very small, fewer than 15 participants. Phase 1, is to identify safe dosages and adverse events. So safety, um, 20 to 80 healthy volunteers or patients, healthy, as in phase 1, healthy, not patients diagnosed with the targeted disease or condition, but safety, healthy. Why? To check for safety. Phase 2, assess drug efficacy. So phase 2 efficacy, assess drug efficacy and refine safety data. And here are several hundred patients with the disease. So bigger sample in compared to the previous phase, and patients actually diagnosed with the condition or with the disease. Phase 3, compare new treatments with standard care or with a placebo. And here, much bigger population or sampling amount or volume, sampling volume, 300 to 3,000 plus randomized participants. Phase 4, as it is in the post-marketing monitor or surveillance, monitor real-world long-term safety, thousands of participants post-approval. Phase-specific monitoring strategies, the phase, the monitoring objective, monitoring technique, key challenges and tools applied. As for phase 0, monitoring objective is centralized oversight of small-scale studies. Phase 1, daily assessments of safety and toxicity, the safety. Phase 2, regular safety and efficacy reviews, so efficacy. Phase 3, consistency across multi-center trials. And phase 4, passive post-approval safety reviews. In table 3 here, monitoring tools and techniques, the tool, the application, the benefit example, and implementation requirements. As tools here, EDC, electronic data capture, or ECRF, IRB, interactive response technology, IRT, interactive response technology. We have predictive analytics, we have the remote monitoring, like virtual monitoring and machine learning. Table 4, challenges and solutions in clinical trial monitoring. The challenge, the impact, the solution, the example, and improvement achieved. As challenges was the that we have discussed are data volume, diverse regulations, limited resources, risk-based monitoring, RBM limitations, and intersite variability. Recent advancements in monitoring, advancement application area, example, benefit, and key tools used. As advancements here, we use that are recent centralized monitoring, machine learning tools, predictive analytics, e-consent integration, and blockchain integration, the audit trail system integration. Application table scenario. For each scenario, the key principle applied, key actions, timeline, and what to watch out for. Evaluating drug behavior, the initial behavior of the drug in phase 0 studies. Key principle applied micro dose pharmacodynamics. Scenario monitoring dose escalation, phase 1 trials, safety and dose escalation. Assessing long-term remission in phase 2 trials, efficacy assessment. Comparing new and standard therapies in phase 3 trials, multi-center data consistency. Conducting post-market surveillance in phase 4, long-term safety and side effects monitoring. Tracking drug allocation across sites globally, investigational product, IP tracking or study medication tracking, SM tracking. Implementing remote monitoring tools in decentralized trials, advanced digital monitoring, addressing high dropout rates in phase 3 trials, predictive risk analytics, ensuring compliance with international regulations, regulatory harmonization. Scenario identifying data inconsistencies in high enrollment sites, key principle applied, centralized monitoring. And next we'll come to some few multiple choice questions, MCQs. We'll start with the question, which key focus distinguishes phase 0 of a clinical trial? Is it A, monitoring rare side effects in a large population? And A would be phase 4, post-marketing monitoring or surveillance. Determining therapeutic equivalence with existing treatment. This would be phase 3, as if we remember. Evaluating drug pharmacokinetics and pharmacodynamics in humans, maybe. D, observing dose-related toxicity in healthy volunteers, that would be phase 2. That would be a phase 1 in healthy volunteers. So mostly will be C, as it is, and it is C as evaluating drug pharmacodynamics and pharmacokinetics in humans. So as A here, rare side effects in a large post-marketing, phase 4, determine therapeutic equivalence with existing treatments or placebo will be phase 3. And D is phase 1. So 4, A is 4, C, A is 4, phase 4, B is phase 3, and D is phase 1. So what's left is C, the pharmacokinetics and pharmacodynamics in the human through microdoses. C for phase 0. Phase 0 trials are exploratory studies, preclinical studies, where a small number of participants, fewer than 15, receive microdoses of a drug to understand how it behaves, initial behavior in the body without therapeutic intent or assessing toxicity. Question 2. What is the primary monitoring activity in phase 2-3 clinical trials? Is it A? Is it A, focusing exclusively on participant safety? And if we remember safety exclusively on safety, if we remember that section or that this purpose particularly is for phase 1. As for phase 0, we said preclinical studies, pharmacodynamics, pharmacokinetics, as we explained here, through microdoses, but using the maximum tolerable dose, determining the maximum tolerable dose, and safety will be phase 1. Blinding and ensuring data consistency across multi-center sites. Well, blinding, masking, uh, and ensuring data consistent across multi-center sites. Mostly, mostly this will be the answer. More probably, C, using large sample sizes to test microdose pharmacokinetics. While we going back, although it's large sample size, large sample volume, but going back to microdose pharmacodynamics will be phase zero still. D, passive monitoring using healthcare feedback. Well, healthcare feedback we know is post-marketing monitoring or surveillance will be four. So the answer here is B, blinding, ensuring data consistency across multi-center sites, as it is for phase three. When we start compare the study medication or the investigational product against placebo or standard of care. So phase three trials involve large-scale comparisons between treatments requiring rigorous monitoring to maintain blinding and ensure data consistency across multiple sites. Question three. What is a common challenge encountered during phase two of a clinical trial? Is it A, determining safe dosage limits for healthy volunteers? Is it B, handling multi-center data variations? Is it C, assessing treatment efficacy without generating vast amounts of data? Is it D, balancing tight resource constraints for frequent site visits? So here we're talking about the challenges, right? So if we remember, one of the challenges is the low budget. So here, will answer will be D, balancing tight resource constraints for frequent site visits. So if we remember, we spoke about the budgeting and using maybe hybrid kind of monitoring in order to consider it more cost efficient, more cost effective. As phase two focuses on treatment efficacy and safety, often generating data across multiple sites. Resource constraints can hinder regular monitoring, necessitating strategies like hybrid monitoring models. Hybrid. Question four, which recent advancement is in monitoring can proactively address participant dropout risks and in clinical trials? Is it A, machine learning algorithms? Is it B, decentralized monitoring tools? Is it C, IRT, which is the interactive response technology for drug inventory tracking, or D, predictive risk analytics? So we are talking about uh, 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 addressing proactively the participant dropout, so retention rates, right, and the risks for that. So mostly will be analysis. So the predictive risk analytics. So predictive risk analytics uses advanced data models to forecast potential challenges such as participant dropout, allowing for proactive measures to ensure trial continuity. Then we have question five. What distinguishes post-marketing phase four clinical trials from earlier phases? Is it A, monitoring initial drug safety in a small participant group? No, we know this applies to phase zero, the preclinical studies. Is it B, detecting long-term effects and rare side effects in large populations, thousands like, for instance, most likely? Is it C, determining dosage escalation and route of administration? No, this wouldn't be phase four for sure, because in phase four, the dosages and the route of administration already have been determined through the prior phases. Is it D, comparing drug efficacy against placebos exclusively? And we know the investigational product or the study medication to be tested compared against placebo or standard of care. This would be phase three. And here we are talking about the phase four, the post-marketing. So it is B, detecting long-term, maybe years effects and rare side effects in thousands, large population, thousands as of sample size or sample volume. So phase four focuses on monitoring drugs after approval, ensuring safety and efficacy over long durations, often covering rare side effects not identified in previous stages. And here we come to the end of this video, this session, or the topic for this video, as which is the first video or first topic in chapter 9. Thank you very much and goodbye.